I
can't
don't
see
see it.
Try
I
to
can't
log
see it.
out and then log in again.
Okay, okay.
Now, can you see it?
You said supposed to be in the group chat, correct?
I posted the PowerPoint in the group chat. Correct.
Okay, good.
But you should see this. You should see the screen because I'm sharing a screen.
Yeah.
It says basic concepts of genetics on the slide.
Yeah.
Can you
I
see
can
that?
see it,
Yeah.
Yes.
All right. I'm just making sure that everyone is joined in. Everyone is here.
Is there a way someone can send me the slideshow? Because I still don't see it in my chat. I don't know if it's just me, but if someone can send that to me.
I'm also going to post this one on the Facebook page. Okay. After the session. Everyone is here. You can hear me clearly. You can see the PowerPoint presentation, right?
Yes.
Yeah.
We're good to go. Someone just left. Are you coming back? All right, let's begin then. Okay. Guten Tag, allerseits. Naimbagamadam kada kayo amin apo. So this afternoon, we're going to talk about the basic concepts of genetics, right? What's a gene? What's a locus? What's allele? Polymorphism, gene frequency. So for this one, know the definition. Okay. Gene, basic unit of genetic information. A locus, we're talking about a location of a gene. An allele, you have two possible DNA sequences at a particular location. So if it's normal, allele, we call it wild type and common. If we have mutations, we call them polymorphisms. So usually mutations are rare. So what is a homozygote? What is a heterozygote? A homozygote is a diploid cell that has same allele at a specific locus on each chromosome. Homozygote, same allele, specific location, locus on each chromosome. Heterozygote, you have two different allele at a given location. Okay, polymorphisms, these are just rare allele occurring in a population with a certain frequency, right? And the genetic frequency is a proportion of a particular allele of a gene within a population. What's high yield here? Know the definition. Next, genotype versus phenotype. And what's the difference? Genotype is the genetic composition of an individual. You cannot see a genotype. You need genetic testing in order to see the genotype. Based on the genotype, you have the following. You have homozygote and you have the heterozygote. What's the difference? Homozygote, you have two homologous chromosomes that contain identical allele at a given location. Heterozygote, two homologous chromosomes that contains a different allele at a given locus. Phenotype, you can observe this one. These are your observable traits of an individual. So they can be dominant or recessive. What's the difference? If a phenotype is dominant, right, it means one of both alleles is phenotypically apparent in a heterozygous individuals. The allele is dominant. So for dominant, only one of those alleles is expressed phenotypically in heterozygous individuals. If the allele does not show up its effect in the phenotype, it is recessive. Okay? So dominant, this is the one that's expressed, the allele that is expressed phenotypically, meaning you can observe this trait of an individual, right? If it is recessive, the allele does not show its effect in the phenotype, meaning you cannot observe the trait of an individual. Codominance, it just means both alleles are expressed in the phenotype. Question, where can we find codominance?
Blood type.
Excellent. So your blood types. So your A, B, and O blood types. So when you think codominance, think about your blood types. What's high yield point here? What's the high yield point? Know the definition. Homozygous versus heterozygous. What do I mean by genotype versus phenotype? Genotype, genetic composition, you cannot see that. Phenotype, you can observe the trait of an individual. The difference between dominant and recessive. Dominant, the alleles are expressed and can be seen phenotypically in a heterozygous individual. Recessive, the allele does not show its effect in the phenotype. You cannot observe the trait if it is recessive. Codominance, it means both alleles are expressed in the phenotype, and this can be seen in your blood types. Oh, another terms that you need to know. You need all of them. You need to memorize all of them. And give me some examples. Penetrance, the probability of individuals with a particular genotype manifesting with the condition. What do I mean if I have incomplete penetrance? There's an autosomal dominant disease. Give me an autosomal dominant disease that you know.
Neurofibromatosis.
Okay, NF1, right? What's the difference between NF1 versus NF2? Which one is bilateral schwannoma? You haven't talked about this? Okay, never mind. I'm sorry. NF2. So, autosomal dominant disease, you can have incomplete penetrance. What do I mean by incomplete penetrance? Anyone?
Sometimes you'll show the phenotype and sometimes you won't show the phenotype.
Exactly. Sometimes it shows the phenotype and sometimes it doesn't show the phenotype. Or in other words, this is a form of penetrance in which not all individuals carrying a gene, a deleterious gene, expressing the associated trait or condition. Okay? That's why you have incomplete penetrance. What's expressivity? Expressivity, the extent of expression of a given genotype at the phenotypic level, the extent of disease manifestation. For example, patients with NF1 have variable disease severity. So different expression of the disease in a different individual at the phenotypic level. They have the same genotype. You have NF1 defect in NF1 gene. However, you express it differently at the phenotypic level. You have different disease manifestation. That is what we call expressivity. Pleiotropy, on the other hand, is you have one gene that contributes to multiple phenotypic effects, meaning it affects multiple organ systems. Examples, phenylketonuria. Can anyone tell me what is this? PKU.
It's when you have an accumulation of phenylalanine leading to brain damage. It's
Exactly. Autosomal what?
Recessive.
Autosomal recessive. Excellent. That's your PKU. What is the enzyme defect in PKU? What is the enzyme that's
That's a
defective
great question.
in PKU?
It's a great question.
So you have phenylalanine hydroxylase. So remember the synthesis of your phenylalanine, from phenylalanine to tyrosine, tyrosine to L-DOPA, L-DOPA to dopamine, dopamine to norepinephrine, norepinephrine to epinephrine. When you think about phenylalanine, think about catecholamine synthesis. Throughout the rest of the term, you're going to learn about these, I think, in the third module or the fourth module, this catecholamine synthesis. What's some sign and symptom of someone who has PKU? You have a musty odor. They love that on the vignette. They really do. If you've seen the baby has a musty odor, think about PKU. Why does the baby have white hair or a form of albinism? Hello? Any volunteer? Did you talk about PKU in class or not yet?
Not yet, like not in detail.
We did, yeah. I mean, we didn't talk about the exact mechanism on which you get white hair, but we did talk about like white hair.
Okay, so you get white hair. Why? Because the enzyme from L-DOPA to melanin, you have an enzyme called tyrosinase. So this tyrosinase will going to be deficient. Therefore, you cannot have melanin. You cannot convert this your DOPA to your melanin. That's why you have white hair. So PKU, think about phenylalanine hydroxylase. That's a deficient enzyme. Term 2, next term, you're going to learn about tetrahydrobiopterin BH4, which is a cofactor. In every hydroxylase reaction, you need to have tetrahydrobiopterin. You also have white hair because you have a deficiency in your tyrosinase. What won't you give to someone who has PKU? You don't want them to what?
They have to have
You give them
a
less
diet
phenylalanine.
without phenylalanine.
One at a time, please.
Less phenylalanine.
Okay. Do you give them artificial sugar?
No.
Why?
They can't have aspartate.
Can you give them aspartame?
No.
Why? Why can't you give someone aspartame? Because it cannot be broken down, okay? You have a dangerous buildup of phenylalanine when a person with PKU eats what? Any protein-rich foods, okay? No milk, no cheese, no nuts, no meat, even grains, bread or pasta, or aspartame, right? Why? You have a buildup of phenylalanine. Remember, it cannot be broken down into your catecholamine, the pathway itself, right? So what happens? You have damage to nerve cells or in the brain. So we have damage to nerve cells in the brain. That's pleiotropy. So PKU, it affects everything. You have a hair, you have musty odor, your skin, right? Next, Marfan Syndrome, another autosomal dominant. What's the defect in Marfan Syndrome?
Like long fingers, like a protruding chest.
Excellent.
The eyes, trouble vision.
Excellent, excellent. Very good, very good. So think about Abraham Lincoln. Abraham Lincoln, I think a historian posited a theory that Abraham Lincoln has Marfan syndrome. So you have long arms, he's very, very tall, so you have long fingers, right? You have Marfan syndrome. Someone says lens dislocation. Where is the lens dislocation in Marfan syndrome? Is it up and out or down and inwards?
I believe up.
Upward.
Okay, very good. You call this one superotemporal displacement of the lens. Supero meaning up, out, temporal, right? Contrast this one next term when you're gonna learn about homocystinuria. The lens dislocation in homocystinuria is down and inwards. Okay, you have an increase in homogenesic acid. And guess what? Where can we find homogenesic acid? In phenylalanine biosynthesis. That pathway from phenylalanine to tyrosine, tyrosine to L-DOPA, L-DOPA to dopamine, dopamine to norepinephrine, and norepinephrine to epinephrine, right? It breaks from tyrosine to homogentisate. You have that pathway. Okay, you're gonna learn more of this in term 2. So that's Marfan syndrome. Think Abraham Lincoln. Okay, how about next? Sickle cell disease. What's sickle cell disease?
Autosomal
Point mutation from
It's a beta codon mutation,
glyphosate
yeah.
to valine.
Okay, so you have a missense mutation. Therefore, you have glutamic acid to valine. So from hydrophilic to hydrophobic amino acid, right? Glutamic acid to valine. That's high yield. That's the explanation of sickle cell disease. Now, if I have a missense mutation, I have a deficient protein. I have a substitution of glutamic acid to valine. What happens now to my red blood cells?
Gain of function polymerization.
Exactly. So in terms of if there is low oxygen or there is stress in my life, my RBCs, they will gonna sickle. And you might ask me, so what if I have sickling of my RBCs? Is that a problem? Yes, of course. Why? What is the organ that removes your RBCs?
Pancreas.
Careful. It recycles the RBCs.
Liver?
Careful. That's a synthesis. Careful. It starts with S.
Spleen?
Spleen. Excellent. So spleen actually removes your RBCs. And you ask me, so what, Nick, if I have sickling, so what? Then this sickled RBCs, they cannot be removed by the spleen. So you can have blockage in the spleen, right? You can have splenomegaly. And in the end, you remove the spleen. You can have splenectomy. And then you are prone to many kinds of diseases, encapsulated diseases. More of this in term 3. Sickle cell disease, think missense mutation. What happens? Substitution of what? Glutamic acid to valine. If there is a stress, if I take a drug, for example, I might have a reaction. It can lead to sickling of my RBCs. That's okay for now. Compound heterozygosity. What is this? You have the same gene, both chromosomes. It is altered to a mutated allele. So the affected gene loses its function despite the mutation not being homozygous. So this is called compound heterozygosity. The next one is high yield, anticipation. When I say anticipation, you say trinucleotide repeat disorders. Okay, what do I mean by anticipation? A disease increases in severity over several generations or manifests earlier with each generation. For example, Huntington disease. What is the trinucleotide repeat in Huntington disease?
CAG.
CAG. What is atrophied in the brain in Huntington's disease? Your caudate nucleus. We're going to learn more, term 2. The caudate nucleus itself atrophies, so you have decrease in acetylcholine and you have decrease in glutamate for your Huntington's disease. So what happens if a father has Huntington disease? What happens to the progeny? What happens to the kid who is affected? The disease may manifest earlier, right? And it is worse to the kid. So that's what we call anticipation. A disease increases in severity over several generations, and it manifests earlier with each succeeding generation. If a father has Huntington's at age 50, the son will gonna have Huntington's at the age 35, and it's much worse than the father's disease. We call that anticipation. Can you give me an example of other trinucleotide repeat disorders that you know?
Myotonic dystrophy, Fragile X.
Myotonic dystrophy. What myotonic dystrophy? They say, what is it? On the vignette, they're gonna say you shake hands and they can't let go. Think myotonic dystrophy. What else?
Fragile X.
Fragile X syndrome. What is deficient in Fragile X? What gene is
like one gene on the X chromosome?
exact? So you have the frataxin gene. The frataxin gene. What's the trinucleotide repeat there? In Fragile X.
CGG.
CGG. So you have cryptorchidism, you have undescended testes, and you have a giant gonads for that one. Another trinucleotide repeat. Is that it? Huntington, myotonic dystrophy, you have frataxin gene. What else?
I think there's Fredrickson ataxia.
Friedreich's ataxia. Excellent. What's the trinucleotide repeat? GAA. GAA for your Friedreich ataxia. So you have the gait is affected, right? So you have cerebellar ataxia. Next term, you're going to learn about the cerebellum, which regulates balance in the body. So you have something wrong with the cerebellum, you are out of balance. So your gait is affected, the way they walk. It's very, very different. You might ask, so Nick, do I need to know the diseases? Yes. Do I need to know the trinucleotide repeats? Yes. Do I need to know which one is autosomal dominant and which one is autosomal recessive? Absolutely. Do I need to know the defect in the gene? Of course. And do I need to know signs and symptoms? Yes. So another high yield point, allelic heterogeneity versus locus heterogeneity. Allelic heterogeneity means you have different mutations in the same allele, resulting in the same phenotype. Same allele, different mutations, same phenotype. Locus heterogeneity, different location, you have the mutations in genes in different locations that has the same phenotype. Okay, so allelic heterogeneity, same allele, you have mutations in the same allele. Locus heterogeneity, different locations, right? You have mutations of genes at different locations. What's the similarity between allelic heterogeneity and locus heterogeneity? You have same phenotype, same observable characteristics. Linkage disequilibrium, you have in a given population of alleles at different loci, they are associated in a non-random fashion. High yield point here is please know the terms, what's the meaning of each, examples of each, the diseases associated with each terms, and know a general understanding of those diseases. Not in depth, but general understanding. Okay? Okay, chromosomal aberrations. High yield again. Sometimes you have abnormal number of copies of your single chromosome. Trisomy and monosomy. Trisomy is very, very important. You have a presence of a triplicate instead of a duplicate number of a particular chromosome or part of a chromosome. How can I know that trisomy is in my patient? The trisomy is exhibited in my patient in terms of genetic analysis. How can I know what kind of genetic analysis am I going to do for trisomies? Is it PCR? Is it
Karyotype.
Excellent. So you need karyotyping. Remember, they are big. You can actually see for trisomy 31, there's an extra chromosome there. Trisomy 21, this is your Down syndrome. Can you give me a patient who has Down syndrome? Signs and symptoms. So you have epicanthial folds. We're going to put in the vignette. What else? Single palmar crease on the palm. There's a one crease there. Usually we have more. What else? Low set ears. We're gonna put that in the vignette. Philtrum, low philtrum, also for trisomy 21. Did you talk about the, in terms of testing for trisomy 21, what happens to the hormones? What happens to inhibin? What happens to beta HCG? What happens to alpha fetoprotein?
No.
No. Okay, never mind. That's for term 2. Trisomy 18, right? So we have trisomy 21, trisomy 18. So trisomy 18, this is your Edwards syndrome. So you can have rocker bottom feet, you can have prominent occiput for trisomy 18. For trisomy 13, trisomy 13, you have your Patau syndrome. Patau syndrome, holoprosencephaly, meaning you have no calvaria, no skull. For holoprosencephaly, you can have cleft lip and cleft palate. Okay, those are your trisomies. Those are important, high yield. Trisomy 21, 18, and 13. How about monosomy? So for monosomy, only a single copy of a chromosome is present. High yield, Turner syndrome, 45XO. Now, guys, give me an example of someone who has Turner syndrome, the signs and symptoms.
Web neck.
Excellent, web neck. That's a good one. Short stature, right? What else?
Is it low fertility?
Low fertility. Why do we have low fertility in Turner syndrome? Why?
Decrease XO because you don't have the both chromosomes for the sex chromosomes.
So you have dysgenesis, right? You have streak ovaries. Your ovaries are not functioning that well. We call that streak ovaries, right? For Turner syndrome. Excellent, very good. Web neck, don't forget that in the vignette. Short stature and infertility due to streak ovaries. Sometimes you have some polysomy. For example, high yield again, Klinefelters. So we need to contrast Turner syndrome from Klinefelter syndrome. So what is Klinefelters? In one word, think about eunuch. A eunuch, someone who has been castrated, right? Klinefelters, think eunuch. So you have a eunuchoid appearance. You have no development of secondary sexual characteristics. Can you also have infertility in Klinefelters? Yes. Can you have gynecomastia, enlargement of breast? In someone who has Klinefelters? Yes. Can it be seen in boys? Only in men for Klinefelters. Turner's is only in women. Okay? Sometimes you have polyploidy. Polyploidy is an entire chromosome set is multiplied. Like triploid, you have 69XXY or 69XXX. This can only be seen in hydatidiform moles, whether it's complete mole or partial mole. More of this in term 2, next term. So know those aneuploidy, abnormal number of chromosomes, trisomies, the different trisomies and characteristics of each trisomy, 21, 18, 13, monosomy, Turner syndrome, 45, XO, Klinefelter, 47, XXY syndrome. You can also have polyploidy, 69, XXY, or 69, XXX. You can also have some structural chromosomal aberrations. You can have deletions, deletion meaning loss of a chromosome, high yield, cri du chat. This is fancy, French. Cry of the cat, cri du chat. Could someone give me signs and symptoms of someone who has cri du chat? S'il vous plaît.
I don't think we looked at that.
Okay, so cry of the cat, go online and check this one. They sound like a cat that's meowing, a high-pitched cry. In the vignette, they're gonna say high-pitched cry. So look online, okay? Deletion, loss of a chromosome segment. Example, cri du chat, cry of the cat. Duplication, you have a duplication of a chromosome segment. You can also have inversion. Inversion of a chromosome segment. Now, the next one is high yield, chromosomal translocation. When you go to term 4, in the future, you're going to learn about the different translocations. You have chromosomal translocation. What do I mean by chromosomal translocation? the relocation of one chromosome segment to another. Why does this happen, right? It can either be balanced or unbalanced. So remember your chromosome, your P arm and Q arm, right? Which one is smaller? Which one is longer?
P is smaller.
P is smaller, Q is longer. So sometimes they switch, right? They switch with the other chromosome. So for balanced translocation, why is it balanced? Because no genetic material is lost or duplicated. So the phenotype, the observed characteristics of an individual is usually normal. Example is your Robertsonian translocation. The P arm will gonna go to the Q to the P, right? What chromosomes are normally involved in balanced translocation? Can you give me the chromosomes that's normally involved in balance translocation, right?
I don't think we really went over that at all yet.
Okay, I think it is, I don't know if this is high yield. So you can either have reciprocal translocation or non-reciprocal translocation. Don't forget your Robertsonian translocation is very important for balanced translocation. Think about 922. Translocation 922, this is your Philadelphia chromosome for your Robertsonian translocation. Sometimes also chromosomes 13 and 14, 13 and 14, sometimes they are involved also in balance translocations. Also, chromosome 21, 21, right? For Down syndrome, it also has some translocation. So 922, don't forget, for now, for your balanced Robertsonian translocation, that's for your CML, chronic myelogenous leukemia. You're gonna learn more in term 4. Sometimes it's unbalanced, right? It means a genetic material is lost or duplicated. So this is where the phenotype, the observed characteristics, is abnormal. We're good for chromosomal translocation. Let's go another high yield, uniparental disomy. Could you give me the diseases that you know that has uniparental disomy? The 2 diseases, happy puppet?
Angelman Syndrome and Prader-Willi Syndrome.
Okay, now listen up. This is high yield. Prader-Willi Syndrome. Which chromosome is deleted in Prader-Willi? Is it the paternal or the maternal?
Paternal.
Paternal. So Prader-Willi starts with P. So paternal chromosome is deleted. So what kind of uniparental disomy is it? If the paternal allele is deleted, right? What kind of allele is accepted? The maternal, correct? So it is a maternal uniparental disomy.
Would you also call that maternal imprinting?
Imprinting is the gene is not expressed, right? So we can say paternal imprinting because the paternal genes are not, they are excised, they are not expressed. Imprinting means you are not going to express, right? That allele. There is no expression of that allele. Since the paternal one is not inherited, so that one is imprinted. Are we clear?
Crystal.
So Prader-Willi paternal allele is not, it's deleted, right? You don't have that paternal. So what are you inheriting? The maternal, correct? So it is a maternal uniparental disomy. Signs and symptoms of someone who has a Prader-Willi. They left this in the vignette. They said, so there's something wrong with the child and he's having abnormal weight gain. So actually, they can have an obese child. So we can have obesity as one of the signs and symptoms of Prader-Willi. How about Angelman syndrome? Angelman syndrome, what is deleted there?
The maternal gene.
So the maternal gene is deleted, right? So what kind of uniparental disomy is it? What kind of uniparental disomy?
Paternal.
Paternal uniparental disomy. So sometimes in the vignette, they will not gonna say happy puppet. They're gonna describe you. Happy puppet. It's an unusual smile, or they say that kid is very excited. She's smiling all the time. They will not gonna give the happy puppet, that term happy puppet, because you already know it's Angelman. They're gonna describe it to you. So for uniparental disomy, know the difference between Prader-Willi and Angelman syndrome. Which alleles are deleted, which one are expressed, right? And signs and symptoms of each. Know the description, okay? So do I need to know heterodisomy and isodisomy? I don't think so. We'll just read it there. Heterodisomy, error in meiosis 1, and for isodisomy, you have an error in meiosis 2. Know the definition, know examples of each, aberrations, right? Know the diseases involved in each of the chromosomal aberrations, know signs and symptoms of each. Not that detailed, but some of the signs and symptoms. Gene mutations, another high yield. So you have differences. Point mutation, only one DNA base pair. Deletion, more base pairs. Insertion, addition of one or more base pairs. Substitution, you have replacement of one base pair with another. High yield, as I've said, trinucleotide repeat expansion. You have increased repetition of base triplets that leads to what? Faulty protein synthesis. So you have different examples, Fragile X, Huntington's. Know the trinucleotide repeat of each. Know the signs and symptoms. Frame shift mutation. Frame shift mutation, there's a shift in the reading frame caused by what? Insertion or deletion of a number of nucleotides, not divisible by 3. Okay? Frame shift mutation, not divisible by 3, which leads to a modified amino acid coding in the gene segments downstream. So it results in what? Shorter or longer protein that have a modified function. Examples, Tay-Sachs disease. Now, could you please tell me signs and symptoms of someone who has Tay-Sachs disease? So you can have, for Tay-Sachs, you can have red.
It's like a red macula in the center of your eye.
Exactly. So red macula in the eye. So this is due to a deficiency in what enzyme? Hexosaminidase, right? Deficiency for your Tay-Sachs disease. This is what population is most commonly affected by Tay-Sachs disease?
Ashkenazi Jews.
Ashkenazi. Why? Remember counter effect, right? Because of counter effects. Term 2,
Can't really hear
learn
you.
about the difference between between Niemann-Pick's and Tay-Sachs disease. We have a red macula in both Tay-Sachs and Niemann-Pick. However, in Tay-Sachs, you don't have hepatomegaly. Okay? Another high yield, Duchenne Muscular Dystrophy. What's the difference between Duchenne's and Becker's? So Duchenne is?
Duchenne is deadly. There's no production of the male.
So in Duchenne Muscular Dystrophy, you have a frame shift mutation for Duchenne. In Becker, you have non-frame shift mutation. Now, can you give me a sign and symptom of someone who has Duchenne Muscular Dystrophy? So they can have, go ahead.
I think like their calves, like the muscles there become more adipose tissue.
Exactly. So for Duchenne Muscular Dystrophy, think about, there's something wrong with the protein. We call this the dystrophin protein. It actually binds when you go into the histology of your muscle fibers, it actually binds your muscle fibers to extracellular matrix via this dystrophin. Okay, this is high yield. Now, if you have a dysfunction in your dystrophin, you have a dysfunction in your muscle fibers. So you cannot support yourself. You can have weakness in your proximal muscles. You cannot support yourself standing up. So they have this sign called Gower's sign. Gower, Gower's sign, that's high yield. They're trying to lift, they use their hands to lift themselves up. Okay, that's a Gower's sign. And also you have a fibrofatty replacement in your gastrocnemius, right? In your legs. So if you see fibrofatty replacement, think about Duchenne muscular dystrophy. Or if you see Gower's sign, think Duchenne muscular dystrophy. This is much more deadlier than Becker's. Why? This is frame shift mutation. Contrast this one with Becker's. It's a non-frame shift mutation, so they can actually survive in Becker's. It's a mild mutation of your dystrophin. Okay? Please know those two. In-frame deletion or insertion, you have deletion or insertion of 369 more base pairs. Silent mutation, you have an altered codon, right? So most mutation results in base pairs in the third position of the codon. We call this one, the third position of the codon, a wobble position. Why do we call it a wobble position? Why is the third position of the codon called a wobble position?
Because most of the time, whenever you have on the third the base, whenever you're pairing your codon with your anticodon, the third base usually has a variability and it will still code for the same amino acid.
Exactly. Any change on the third base, right, will still code for the same amino acids. So silent mutation, you alter the codon, specifically the third position of the codon, the wobble position, you still have the same amino acid. Nonsense mutation is a formation of a stop codon. So if you have a stop codon, what happens? You have an alteration of splicing and you have termination of translation. Missense mutation, we talked about sickle cell disease, glutamic acid to valine, you have an altered codon. Different codon, different amino acid. Splice mutation, this is alterations in your nucleotide sequence, your SNRPins, right? The formation of your spliceosome. There's something wrong with the formation of the spliceosome on the exon intron. So the introns are there, they are not spliced. Okay, therefore you have a defective mRNA and you have a shortened protein. Example of splice mutation, beta thalassemia. What's high yield point? Know the definition of each. Know the diseases associated with each gene mutations. Know the signs and symptoms associated with each one of them. Okay? Did you talk about beta thalassemia or not yet?
We did.
What's the difference between beta thalassemia major and minor?
I don't think we got that in depth.
Okay. Okay, never mind then.
We literally just made the connection between splicing and beta thalassemia, and that was it.
Okay, okay. Probably in the future, right? So there are different kinds. Beta thalassemia, you have major, beta thalassemia minor, right? Contrast also this one with alpha thalassemia. We're going to learn more in the future. Okay, guys, so this is from first aid. Know the term, know the definition, know the examples, okay? And they might give you the signs and symptoms of that disease, and then they're going to tell you what genetic term is associated with that one, okay? So if you know the example, know some signs and symptoms of each. NF1, BRCA, PKU, trinucleotide repeat, right? HNPCC, leave from many, right? And you have dominant negative. Mosaicism is high yield. So in mosaicism, you have genetically distinct cell lines in the same individual. The difference between somatic and gonadal mosaicism, somatic, it arises from mitotic errors after fertilization. This one, it propagates from multiple tissues. Gonadal mosaicism only in the egg or sperm cells, only in the gametes, right? So in the vignette, if the parent and relatives do not have a disease, you suspect germline mosaicism. A disease associated with this, McCune-Albright syndrome, okay? And we talk about locus heterogeneity. Example of this one is albinism, right? You have loss of tyrosinase for your albinism, allelic, beta thalassemia. Okay, this is another high yield, heteroplasmy. So when you think about mitochondrial disorders, think about heteroplasmy. Okay, it means you have a normal and mutated mtDNA, mitochondrial DNA, resulting in what? Variable expression. Don't forget that it is passed from mother to all of the children, meaning all of the children will going to have that disease because we only inherit mitochondria from our mom. And we talk about uniparental disomy. Okay, here is high yield again. Linkage, no, this is pedigree analysis. You need to know the linkage analysis. They're just gonna give you something like this, and then they're gonna ask example of autosomal dominant inheritance. So you need to know how to analyze pedigree. Okay, let's begin here. So there are different kinds of symbols, right? For family members, you have square for male, circle for female, horizontal line, marriage, right? Vertical line, progenies. Okay? If you have
we can't see what you're referring to.
Come again?
We can't see. We're only seeing the PowerPoint that you have, but we can't see like the pedigrees you're talking about.
Oh, so this is a pedigree analysis, right? So you have different kinds of symbols. So how am I going to do pedigree analysis? First, check, is it dominant or recessive? How do I know if it's dominant or recessive? Does every affected family members have a parent that is also affected? Does every affected family member have a parent that is also affected? If yes, dominant. If no, recessive. That's the first thing that you need to do. Next, is it autosomal or not? So your second question should be, are male family members mainly affected? If yes, then it's X-linked recessive inheritance. If the following criteria are met, right? The disease mostly likely follows an X-linked dominant. How do I know it's X-linked dominant? Do all affected males also have an affected mother? Do all affected males have healthy sons? Does no affected male have a healthy daughter? Then you think about X-linked dominant. If one of these criteria are not met and question 1 has been answered in the affirmative, the disorder is most likely autosomal dominant. However, if question 1 is answered with a negative response, the disorder is most likely autosomal recessive. Pedigree analysis, what do I need to do? First, I need to determine, is it dominant or recessive? Does every affected family member have a parent that is affected? Yes, dominant. No, recessive. Next, are male family members mainly affected? If yes, X-linked recessive. How do I know that it's an X-linked dominant? Do all of the affected males have an affected mother? If yes, X-linked dominant. Do all affected males have healthy sons? If yes, X-linked dominant. Does no affected male have a healthy daughter? Then yes, it's an X-linked dominant. Now, we're gonna go over some exercises, okay? Look at this pedigree. Using that analysis, look at the pedigree. What do you think?
I still cannot see the pedigree you're referring to. Like, for some reason, the screen is still stuck at linkage disequilibrium for me. I was wondering, is that the case for everybody else, or?
Yeah, you can look also in the chat. Someone is like screenshotting them and putting them on the chat.
Oh, is there something wrong with Skype?
Maybe it's frozen.
Do I need to log out and then log in again, or?
You can try.
Now I can see it.
Oh, you can see it?
Yeah.
Okay, there's a pedigree here. Can you all see it?
No.
I logged in and out, guys. So just log out and log in again, and you'll see it.